Okay, so we're back with another episode of Mason's Medicinals for the Real Mushrooms interview series. And today we have a very, very special guest, Dr. Kara Fitzgerald. And Dr. Kara Fitzgerald is a phenomenal clinician. She has a very, very interesting background. So, welcome to the show, Kara. I'm just going to give a little bit of an intro to Kara here. And she's a naturopathic doctor and a functional medicine certified practitioner, and she was actually the recipient of the first ever Emerging Leadership Award from the Personalized Lifestyle Medicine Institute in recognition for her work in the area of DNA methylation. And as a leading voice in this epigenetic DNA methylation area, Dr. Kara has been working on this for a big part of her career, and she's a certified expert in the area. She has also been on the faculty or she is on the faculty of the Institute for Functional Medicine, and she has a clinical practice in Newton, Connecticut, where she lives with her daughter. She's got an excellent new book releasing at the end of this month, and there is some really, really good reviews on it. And we'll get into her book today, and it's called Younger You, and it's all about reversing your sort of biological age to help you live a better life, stay younger, and work on this epigenetic concept that she's been working on for many, many years. So welcome, Kara.
Thank you. It's great to be here with you.
Yeah, we're really excited. So why don't you just tell the group what your interest is in health, how you kind of got where you are today, what was that kind of aha moment for you, where you wanted to kind of go into this line of work, because not everybody gets into this line of work.
Now, do you mean naturopathic medicine as a whole or focusing on laboratory science and getting into epigenetics? Like, how far do you want me to go back?
The epigenetics and the laboratory studies and start there.
Yeah, sure. Okay, well, I knew way back as a student in school, when I went to the National University of Natural Medicine in Portland, Oregon. And of course, Dr. Jeff Bland, the father of functional medicine, is there in the Pacific Northwest. And I was able to see him lecture. And as soon as I was exposed to nutritional biochemistry, it resonated with me deeply, deeply. And I knew that I would be using my degree beyond clinical work, that a portion of my time would be spent seeing patients, but that a portion of my time would be spent in a laboratory. At least that's what I envisioned. And initially, I thought that, I might go back for a PhD. But then when it came time for me to think about my next journey upon graduation, I decided to apply for residencies, and lo and behold, discovered that Metametrics Laboratory, now part of Genova, had a postdoctorate position in laboratory science under the direction of Dr. Richard Lord, who's a very highly regarded nutritional biochemist. I couldn't have made that up. I mean, I couldn't have dreamed a more perfect position to be really mentored, at the feet of a master, from the just graduating, from the first steps into my career, to be so steeped in laboratory science. While I was there, of course, we launched the first PCR, DNA stool analysis in the clinical setting, in the clinical laboratory setting. We did a lot of firsts. We did a lot of laboratory investigations that were pretty cutting edge. And so I was comfortable swimming in that pond. So that's my background. And plus, I was a clinician. And then I finished that postdoctorate position and hung out my shingle here in Connecticut, where I'm from. So one of the things I took from the laboratory was having a community of like-minded thinkers with me. Like it really takes a village to be a good physician, to be a good scientist. You need to have a sounding board. And so after I left the lab, where we had a rich think tank of clinicians and scientists, I needed to recreate that. It was a big loss when my time there was completed. And so I started a journal club. I recommend all of us think about that. So if clinicians are listening, we tend to get siloed. And how do we keep our community going? How do we stay interconnected? How do we keep each other current? How do we translate the science into clinical practice? All of those really important questions were up for me. And the first thing that I did was create a journal club of my friends who wanted to, on a monthly basis, explore topics, explore, research studies, talk about patients, et cetera. We did that. And then eventually my clinic here expanded. We grew it. We started a nutrition internship program. We started to have rounds that were fairly large. There's one, two, three, four, there's five, six physicians here now and about 10 nutritionists because we have a nutrition internship program. And we've got lively, good rounds. We've got a Facebook structure, a private group where we, dialogue and keep each other current. Since made that into something called clinic immersion, where we invite clinicians from the outside space to come in. So very important to me is staying connected to my colleagues. And that just keeps me in the literature, keeps me in the science, and it just, keeps me thinking ahead about how we interpret, just what are our next steps as we move into sort of this omics era where we can see gene expression. We can just see so much. Laboratory science has evolved. I don't want to digress too far. In around 2013, I began to really tussle with the literature on epigenetics. What struck me was the bulk of that literature was coming out in oncology, and the tumor microenvironment takes over gene expression, and particularly DNA methylation. Very simply, a methyl group, carbon and 3 hydrogens, when there are a lot of methyl groups on a promoter region of a gene, generally that gene is turned off. Conversely, when those groups are removed, that gene is on. Cancer, kind of the tumor microenvironment sort of wipes off a lot of methylation. So it turns on genes willy-nilly, but then it shuts off almost important genes that we want on, genes that protect us. So what happens is this kind of disorder of hyper and hypomethylated gene patterns. That same kind of occurrence happens in all of the chronic diseases of aging and in aging itself. So as I metabolized this, I wondered, what are we doing? What are our interventions doing? Are the interventions in functional and naturopathic medicine always supportive of the best genetic expression? Or could we be pushing things forward unnecessarily so? Could we be contributing to imbalance? And that became a huge question for me. Sometimes I think if we're dosing up excessively on isolated B vitamins, it may not always be the best idea. And so to that end, in our practice in 2016, well, even before then, we created a diet and lifestyle program around optimizing DNA methylation. Nowhere in the literature is there any suggestion that excess leafy greens is going to cause cancer, right? It's all the reverse. In fact, if anything, it's protective. And so we put together a program that we thought would optimize DNA methylation, protect us from some of these diseases of aging, and be able to offset our need to rely on isolated vitamins. I prescribe vitamins all the time as a physician, but now I tend to go a little bit lower dose. I might go for a shorter duration. So we created this program, flat forward to 2017. So we actually released the program as an ebook. I lectured on the program at IFM, and I actually lectured on it, around the world. Other functional clinicians were interested in what we were thinking about and starting to think about epigenetics. But a niggling question was, are we actually changing epigenetics? And at the time, you can't send somebody off to, Quest Laboratory for an epigenetic workup. They're just not available to clinicians. And so I was given an unrestricted grant by Metagenics to study it. And we hired the Health Gut Research Institute at my alma mater, NUNM, to run the study. And it was an 8-week-long intervention. We looked at middle-aged men. Middle age is when DNA methylation tends to get wonky. Genetic expression tends to grow wonkier with age. And so that was the population that we wanted to look at. And Metagenics gave us, the funding in order to do it. Extraordinarily, and this was beyond our expectation, at the end of the 8 weeks, as compared to our control group, we saw that we reversed biological age using a DNA methylation clock, which we can talk about, but we saw that we reversed biological age by over 3 years on our study participants. And this was unprecedented. This hadn't been demonstrated before. And as a result, our study got quite a bit of attention. I just want to touch on the fact that aging, so biological aging, is the biggest risk factor for the chronic diseases, for cancer, for dementia, for cardiovascular disease, for diabetes. And so reversing biological age should, by extension, reduce risk for these diseases. And what I have observed in my read on the literature is, as I said earlier, when you look at epigenetic expression, aging actually looks a lot like these diseases. Like you see the same sort of deterioration happening um, on the epigenome. So it was, it was a pretty extraordinary finding, and that was in 2019. Actually, at McGill, I was working with Moshe Sef at McGill University, and Josh Middledorf was our biostatistician helping with our clock analysis, and that was just before COVID hit. And then shortly thereafter, I was asked to actually write the program up for consumers, and that's the Younger You book that you mentioned. So there's a long-winded introduction to what I'm doing, and now I just want to stop. And
Thanks so much for going into how you got into this and why it's so fascinating and so interesting, and how the stars kind of align just with that postdoc nutritional biochem, and then going out into the research world, and then yeah, doing active studies, being an active clinician. It's so fascinating to hear, and all this new epigenetic talk can be, it can be confusing sometimes, but when we get an expert like you, it's really good because you can kind of distill it down. So what about, you said, in this 8-week study, you're able to turn around the DNA methylation clock. So for listeners that are kind of familiar with epigenetics, what is the kind of micro expression of it? It's at the epigenome level, you said, and then how does that trickle out into our bodies? Like, what organs does it affect? What cells? Like, what's the through line there?
I'm going to back up. Mason, don't hesitate to interrupt me if I'm not clear in any way, or I need to restate something or whatever. We mapped the genome out. The human genome was mapped in the early 2000s, and we have a lot less genes than we anticipated. We've got like 23,000 genes, and there was an expectation prior to mapping it that we'd answer a lot of our questions around chronic diseases, that we would have like a genetic answer for why these are occurring. In fact, we didn't. There isn't a clear through line from genes to chronic disease. And that really exploded the era of epigenetics because we saw that it was above the gene. So technically, the term epigenetic means epi, above, genetics, the genetic material, so above the gene. So rather than our genes being the cause of accelerated aging or longevity or our risk for cardiovascular disease or cancer, et cetera, it's not. It's what is above the gene. And that is the interface of environment. So that is how we live our life. So how we live our lives, and we can talk about there's a heritability component in epigenetics as well, we can talk about in a minute, but how we live our lives is filtered here, influences gene expression at the level of epigenetics. There are a number of different biochemical processes that happen that regulate gene expression. The best studied, and really arguably one of the most important, is DNA methylation. One of the reasons it's the best studied is because we have really good tools to measure DNA methylation, whereas acetylation and changes to the histones, the proteins that the DNA is wrapped around, like we don't have quite the same, or looking at small RNA and their role in gene expression, we don't have quite the same level of reliable tools to investigate the other epigenetic processes. But DNA methylation is special for a few reasons. We can preserve DNA methylation patterns, cell division after cell division after cell division. There's enzymes in the body, DNA methyltransferases, that keep those patterns intact, which is not true with some of the other epigenetic marks. DNA methylation and demethylation, they determine the fate of pluripotent stem cells. So in embryogenesis, methylation is largely wiped clean, not completely, and that's the heritability component we can talk about, but mostly wiped clean. And then that embryo's development occurs via methylation pattern. Is this pluripotent stem cell going to, turn into an optic neuron, or is it going to be a skin cell, or is it going to be a heart cell? It's extraordinary how powerful it is, and it's consistent. So those marks are preserved. Like you want your skin cell to remain a skin cell, right? You don't want it to turn into a liver cell or something. So those marks are resilient and preserved over time. One X chromosome in those of us with two X chromosomes is methylated to shut it off. And that is clearly preserved over time, over, cell division after cell division. So it plays methylation and demethylation play very fundamental, extremely important and long-lasting roles in the body. It's also very responsive. So other regions of the methylome, as it's referred to, are responding to our daily inputs. And there's a continuum. So depending on, the quality of our inputs, it will dictate, gene expression's fate for the better or for the worse. So that is a little bit of background. And let me see if you've got any questions or if I've answered yours.
Yeah, that's great. I just wanted to take that further now, and if you don't mind, just going into, there's a lot of information out there, even in this kind of arena. And what are some of the big kind of myths or mistakes that you see people making when, using this information from a lifestyle perspective or a diet perspective or a supplement perspective? What are the big kind of myths that you found when working through this info?
When SNP testing became widely available, clinicians were ordering it, and then, consumers can get, a massive amount of genetic data. And that data often comes with interpretation. And, if you've got this SNP, you've got increased risk for this, this, and this, and therefore you should take all of these supplements. I think that there's just gross overinterpretation out there. And people get really anxious about their SNPs. I think the promise of epigenetics is that your genes don't dictate your fate, or very limitedly so. All of us have had patients come to our office with a really high degree of anxiety because they've done SNP testing and they think that they need to be taking supplements X, Y, and Z forever, or that they're going to get diseases X, Y, and Z. I talk about some of that in my book. I think that was originally kind of speaking to that, like, let's maybe course correct the conversation. So that's one piece. But the other piece is, in this era of being able to see gene expression, we're starting to be able to see gene expression. Believe me, in another 5 to 10 years, we're going to have more tools. We can see how our diet and lifestyle, our nutrient inputs, our stress level. I mean, we can see how this influences genetic expression. People probably have heard of the agouti mice studies. Randy Girdle and Waterland, in the early 2000s, published the first extraordinary nutrition epigenetic study where they looked at an agouti mice, or a group of agouti mice, pregnant agouti dams, and they're obese and blonde, and they're vulnerable to diabetes and cardiovascular disease because they have this this agouti gene on. So they're very visually distinctive. They look like these little orange blobs. And Girdle and Waterland gave them methyl donors. They gave them B12 and folate and betaine, and lo and behold, they were able to methylate and turn that gene off and really restore the mice to like a wild type. They call them pseudo agouti. They're, skinny little brown, house mice after that. So you can see the power of these nutrients influencing genetic expression ridiculously potent in this study. And I want to point out that those changes in that first generation lasted multiple generations. So it wasn't just the immediate offspring. Those vitamins had impactful results on genetic expression generations out. That's a big deal. I mean, if that doesn't cause you to think about how you're dosing your vitamins, we're not just peeing them out. They're not harmless. Like I was taught that they're water soluble. They're not a big deal. You're not going to store them. You're going to get rid of anything you don't need. Yeah, maybe. And they're also influencing gene expression. They're potent. Waterland and Girdle summed up their abstract in that first paper, and actually subsequent papers they would talk about this, cautioning our understanding of nutrition supplements, basically suggesting that we recognize the power of them to influence genetic expression, and that we be mindful of that. I think that our focus and obsession on single nucleotide polymorphisms and prescribing shopping lists of supplements to sort of remedy the failing of our genes was just really kind of misguided in our approach. And so, again, we created this very, very food forward approach, recognizing the potential to influence gene expression. I wasn't sure that we would reverse biological aging in our study. Obviously, I hoped that we would. It was one of the principal things that we wanted to look at. But I felt certain, maybe it's naivete, but I did feel certain that we would influence genetic expression. I felt certain that we would see some changing to the epigenome, and we have, and we'll continue to unpack and publish on our findings. But the first big finding was that we looked at the genes involved, or the methylation patterns involved in biological aging, and we took them to a younger pattern, which is pretty extraordinary. I think it's cool for a lot of reasons, but it certainly highlights the power of diet and lifestyle. We didn't use any supplements. We did a greens powder and we gave a probiotic. So a polyphenol concentrate and Lactobacillus plantarum.
In your book, you go through a lot of these lifestyle nutrition guidelines to help, with this biological age concept. So I know you talk a lot about a couple key foods and a couple key lifestyle practices. So can you run us through that based on your research and what you found?
We took principles of naturopathic medicine. In fact, I did a podcast with Tina Kaiser, who's just a really amazing naturopathic doctor, and she's part of the Natural Medicine Journal. She said, I read your diet, Kara, and I just cracked up because this is old school nature cure. This is an old school nature cure diet. And I said, yeah, it really is. It is. And she just got the rationale for everything we were doing. But I want to tease out that we designed the diet heavy in methyl donors, again, knowing that methyl donors delivered in a food matrix are really uniformly safe. At worst, if you look at the research, they're neutral. They're not impactful, but really uniformly, a methyl donor heavy diet is very safe. And we know as we age, methylation gets wonky, as I explained, hyper and hypo are happening, but there's a net loss of methylation. So a global hypomethylation pattern, and then areas of hyper and imbalance. So it's just messy. It's messy in the diseases of aging as well as aging itself. So the diet needs to be loaded with methyl donors, loaded. We use the liver, which is a nature cure, old school nature cure ingredient, and it's a multivitamin in a food matrix. In addition to it being methyl donor dense, the new research on polyphenols is that they're epinutrients also. So they're not methyl donors. They're not pushing methylation forward, but they influence the enzymes. And it seems like what they're doing in part is directing where methylation is happening to a more favorable pattern. I write about in the book, there's actually a table in the book, and most of this is in vitro, some animal data, so it's new, but looking at tumor suppressor genes. So tumor suppressor genes are routinely hypermethylated and turned off in cancer, as I said, and also in aging. And there's a pretty decent body of literature in vitro and animal showing these beautiful polyphenols allowing these tumor suppressor genes to be turned back on. And this is beyond just tumor suppressor genes. I focus on that because that's where the bulk of the literature is. They seem to support good genes in general sort of being turned on. And I'm talking about curcumin and the catechins in green tea, or resveratrol, luteolin, quercetin, rosmarinic acid in rosemary, the polyphenols in blueberries, and on and on and on. There's so many of these epinutrient, these epi-active polyphenols. I built out a massive appendix. It's like a 30-page appendix in the back of the book where I talk about epinutrients, including donors, as well as this category of polyphenols we're calling methylation adaptogens. And then there are demethylation. So there's a whole nother family of enzymes called 1011 translocation enzymes that actively demethylated a gene and allow it to turn back on. And there's nutrients associated with those enzymes as well. And so I just list them all in food sources in the back of the book, which I just think is cool because you can see how you can eat for your genes. You can eat for this optimal gene expression and do it routinely and easily. So we designed the diet around that. It is old school nature cure, but we're thinking about DNA methylation, and it's designed like orthomolecular principles, so the right amount and the right quantity to really move DNA methylation. So we really leaned on orthomolecular science, on functional science, on naturopathic medicine. Now, beyond thinking about the methylation cycle, we see that there are many other inputs that influence DNA methylation. Exercise is a potent influencer of, well, we know it's favorable in longevity. I mean, there is too much exercise, and I get into that in the book. So excess exercise could be a pro-aging event. But, balanced exercise, which can be pretty intense, I mean, it depends on your level of fitness, but it doesn't have to be, it's routinely favorable. And it's favorable epigenetically. A cool recent study shows exercise behaving like these polyphenols I just talked about, turning on previously inhibited tumor suppressor genes. Another study speaks to exercise's benefits as being mediated largely through changing genetic expression at the epigenome. So we see the downstream anti-inflammatory effects, the mitochondrial benefits, the antioxidant benefit, all of the myriad benefits of exercise, it's now being theorized, are sourced to gene expression. So exercise was a no-brainer that we wanted something in there. And we did a modest prescription of 30 minutes, 5 days a week of a perceived exertion, 60 to 80%. So not intense, and you could do whatever you want. We didn't limit what people's choices were. A lot of the guys in our study walked. Sleep is really important for epigenetic expression, and I get into that. And we wanted our participants to hit, a minimum of 7 hours. Little sleep, especially in animal studies where they can, do a brain analysis, a brain dissection, has an immediate and impactful effect on neurogenesis. So sufficient sleep is essential for DNA methylation and epigenetic regulation. Stress, I would say that stress drives aging forward stronger, as strong as anything, and maybe it'll, prove to be one of the top drivers of aging. And an aha moment for me of the many was the biological age clock that we used in our study, which is sort of the original kind of flagship, best published biological age clock. A full 25% of it is devoted to influence by glucocorticoids. So you could say that stress influences 25% of this particular biological age clock. So we prescribed a meditation program, just a very simple relaxation response by Herbert Benson. It's a validated, tool. He was at Harvard, but just a simple breathing exercise. And we wanted folks to do it 10 minutes twice a day. The data on meditation, on yoga, on Tai Chi, is just really cool. And it's still a new area. All of epigenetics is sort of a new area of investigation, and the science will continue to evolve. But you see how potently anti-aging it is. Especially in experienced meditators. But even so, the cool thing about this is even one meditation and exercise, we can talk about exercise too, even one exercise event can have favorable changes. Obviously, they sort of spread like wildfire and benefit if you continue to engage in the practice. So exercise, meditation, sleep were the lifestyle pieces.
Yeah, it's all coming back to the nature cure with the diet and the modification factors. And so those are the things that help, at this epigenetic level. These are the things that we want to try to support in our everyday life. What are some big things that negatively affect these pathways that you've been speaking about for the last little bit? And, you mentioned lack of sleep, but, what are some other bigger areas that you've seen with the research?
It's kind of interesting. It was Sid Baker, another, granddaddy in functional medicine, had this idea that he called get rid. So what do you need to get and what do you need to get rid of? And I'm talking about a lot of what we're getting with our study interventions, but what are we getting rid of? So processed food, sugar, what else? Stress, poor sleep, no exercise or excessive exercise. All of these things have negative influence on gene expression. So we couldn't make our study organic, or we would have had to supply. It was expensive. Clinical research is not a cheap adventure, and I'm again just so deeply grateful that Metagenics took this on, because it's a big investment. It's a big investment in time, but toxins are, just bad actors as far as, well, not just in gene expression, so in DNA methylation, but also just in, the quality of DNA and DNA damage. Toxins, uniformly are quite disruptive. So while we couldn't prescribe an organic diet to our participants, we encouraged them to eat as cleanly as possible. What else? Avoid using plastic. Just if you can't buy organic, making sure things are appropriately soaked in vinegar and rinsed and washed and so forth. And since we prescribe liver, we wanted people to get as clean source liver as possible. I know that was a big dialogue. We had our nutritionists work with each of the participants on a weekly basis, and that was always an active dialogue where people could find clean sourced liver in our study. So I think that's a very important piece, and one that I spend a lot of time in the book teasing out.
Great. So we'll find that info in the book for sure. A couple other concepts. So vitamin D, and we hear that vitamin D is a molecule or a hormone that is associated with a lot of these different pathways. So what's your kind of short education for us kind of newbies to this area on how vitamin D is interacting with all these things you're talking about?
Would just say yes. You know how it is with vitamin D, right? It just influences everything. Yeah, it's important. We didn't prescribe vitamin D in our study, but it's important. It regulates a lot of epigenetic processes, not limited to DNA methylation. It's extremely important. In fact, a paper that came out before ours went to press looked at obese, vitamin D deficient African Americans, and they measured biological age in this population. And the group that received 4,000 IU, because they had different dosing structures in it, reversed their biological age in 16 weeks by 1.85 years with just giving vitamin D. So I think that, it's not as, it's not as splashy as what we saw in our study in 8 weeks, over 3 years, but a single nutrient in vitamin D deficient individuals yielded, almost 2 years age reversal. So I think that really illustrates the incredible importance of vitamin D in gene expression. And it does it via a variety of mechanisms. I mention it in the book. There's a supplement section in the book, by the way, and I actually, just in case I forgot, I'll just say it right now. I talk about mushrooms. I think mushrooms are, I think that we'll appreciate their influence on epigenetics, on gene expression in time to come. There hasn't been that much research on it, but there is some out there, and I have absolutely, well, they're nutrient dense with methyl donors, and there are some compounds that have sort of a methylation adaptogen influence, but I do mention them in my book, in particular, like the fruiting body, like the real mushroom. But I do think vitamin D is an important nutrient for us to be using, and we allowed our participants to take vitamin D had they already been, although not very high doses. So we did control that.
Okay, so that's the vitamin D scoop. Yeah, so a lot of these herbs and things rich in polyphenols, some could be classified as adaptogens, I guess, depending on your definition.
Our definition is loose. It's when I'm talking to a population that's educated in this arena, it doesn't fit the strict definition of adaptogen.
I figured, and I feel the same way. They're amazing, all in their breadth of use.
With the donors, like I think as a diet as a whole, they could kind of sneak into the methylation adaptogen definition, but yeah, they're not technically.
Okay, so that's vitamin D. We hear eating the rainbow. Like, what's going on there with like these pigments and these? You mentioned blueberries, you mentioned like cabbage in your book, and you've got a lot of pigments in some of these very powerful foods. How do these high pigment foods And they're most of them are methyl donors, right?
They're not necessarily, no, I would say they're mostly what we're calling adaptogens. Um, there are some of them do have donors, but really mostly they're epinutrients in that they influence the behavior of enzymes, um, of the methyltransferase enzymes, and seem to support expression of genes we want on. Perhaps we can use adaptogenic in that, in that effect. They don't just willy nilly turn on genes. They do not. They do it with some intelligence. My strong suspicion is that we know these pigment-rich foods. We know these polyphenols from time immemorial, like green tea, curcumin, some of those long, long use history nutrients are anti-inflammatory, they're antioxidant, they're detoxing, they're anti-cancer, they're antimicrobial. They just have these rich complement of effects in the body, very far reaching. And I suspect we're going to see, that this is sourced back to the genes that they support being on and the genes that they support being off. I mean, they seem intelligent. So I'll give you a description of this, an example. In our study, we didn't increase net methylation in our participants. We measured homocysteine, S-adenosine, methionine, et cetera, et cetera. We measured a lot of the methylation cycle markers, and none of those changed appreciably. We didn't increase it, but what we did was we repositioned methylation in a more favorable pattern with what we did. We moved it around. We moved what genes were on and off via DNA methylation. That's what we showed. I just think it's really cool.
Super cool. Yeah, it really is. So I want to ask you about a couple other things that are kind of in line, but looking at, there's animal proteins that are suggested, there's plant proteins, there's, the macronutrient ratios. In terms of all that stuff, how does, macronutrient ranges kind of play into this? And then also, animal foods, there's these big trends for like, pretty strong animal favored foods. And then, other people are still working with, pretty low, minimally added animal foods. So macronutrient ratio, what's your take on that? And then also the balance of the actual diet. What's important
It is we wanted to lower glycemic cycling. We wanted to keep blood sugar modest. We wanted it to be keto leaning. I think ketones are favorable epigenetic modulators. Um, so to that end, the fat is a little bit higher. Uh, Carbohydrates, it's a grain-free diet. We wanted to keep it anti-inflammatory. So, things that we got rid of, going back to that, I talked about toxins before, but it's an anti-inflammatory diet as well. Grains are out, legumes are out. It's a dairy-free diet. There's no alcohol. So more things that I pulled out. Um, Alcohol inhibits methylation, actually some of the methylation cycle enzymes. And this is not a forever diet, or it can be if you want it to be. Like, it is the eating pattern that I stick with most frequently, but one needn't give up alcohol or grain or legumes, actually, which are really good nutrients forever, just for this period, for the 8-week period. So our macros are in keeping with those goals, with wanting to be keto leaning. Protein requirements, they're not crazy high. We do recommend in the book, I recommend more protein as we get older, more protein in pregnancy, more protein for people working on building muscle mass. Protein can even work if you're sort of detoxing from sugar. It can kind of help with cravings. So there are appropriate times to kind of use extra protein, but it's not a huge animal protein forward diet. It's definitely a plant forward intervention more than anything. But I absolutely appreciate the nutritional power of well prescribed animal protein. In that we have an everyday version, which we haven't studied. It's in the book. It's called the Every Younger You Every Day, and That allows for some grain. It allows for a little bit of dairy if you wanted. It allows for alcohol. I don't know if that's going to reverse bioage because, like I said, we haven't studied it, but it's a doable program that we used quite a bit in clinic practice. Um, and so I'm outlining it in the book. So you had your question on macros, and then you had another question.
I think that covers it. My next one, though, if you're okay for another question, is you're still doing your journal clubs, your little think tanks. You're always getting in different scenarios where yeah, I liked what you said at your intro. You're always trying to, mix it up and get in with some different minds so that you can see what's coming next or see what's in different areas. So what do you think is kind of your next step here or the next kind of phase of this evolution of this understanding of epigenetics? It's a very loose question, but yeah, where do you see things going from here?
I want to say just in terms of me mixing it up and keeping, being close to other people who are drawn to this field and passionate about it, I like to support folks in, reaching for their best. And just because that was offered to me, when I was a new graduate and doing my training. And it just feels very seamless for me to continue to keep that. And I just appreciate it when I see new clinicians come on board here or a new nutritionist, just reach higher and dig deeper. I mean, that's something that gives me very deep satisfaction and it helps me do the same. So that's that piece. Um, if we're going to change, the trajectory of health in our countries, our respective countries in the world, I mean, we've got a lot of work to do. We need everyone, all hands on deck. So I just like to support, um, this generation, the next generations to come in that very fundamental mission. For me, in terms of epigenetics, I'm really excited about the power of it and the fact that these tools are becoming more and more available. We have an app, we have a digital program. If people go to younger-you-program.com, they'll see the book, they can order the book if they haven't. And then just below that is our digital platform. It's called 3YY for 3 years younger. The digital platform has everything to jump into doing the program that we used in our study, right down to the nutritionists that we used. But the epigenetic testing, the supplements, et cetera, it's all in the digital program. We have IRB approval to continue to study it. So there's a research arm going on as well. My vision is what I've been working on is designing an affordable epigenetic array so that we can use this widely. So that's what I'm working on. We have to just hold the energy that I can make it happen, and we'll just, everybody can get their biological age clock and then some. Really is a couple times a year, I think would be ideal to make sure they're doing what they need to do to stay optimal. So I want to continue to unpack this science. I think what the other piece that I would like to do in the app is look at, what do we want to stack on younger you? So we have this foundational intervention, but I was podcasting earlier with Mark Hyman, and he was saying, yeah, but what if we really individualize it? Then what's happened? What if we see it through the systems lens and really address the individual in front of us and not just this? So this is the foundational intervention, but then let's layer on, dealing with their toxic burden, dealing with their micronutrient deficiencies, and just really optimize through the functional lens. What kind of bioage reversal are we going to see then? And of course, there's all sorts of different nutrients that are being established as senolytic agents, as longevity agents, and what if we layer in some of those? So I think it's an exciting time, and it has been for a long time because we're in this systems biology paradigm now, and some of the tools from systems biology are more available to us as clinicians, and so I think we can do some pretty cool stuff, and I would like to participate in answering some of those questions. So that's my fantasy.
Awesome. It's wonderful to hear. So I always like to ask people, obviously you're doing some of these things that we've mentioned, but, from your own personal, not even health, but your own like life perspective, like what are some of the big things that you try to just put as your foundation to keep you feeling, happy, healthy, but also just feeling, grounded, centered?
Well, I've got a toddler at home. She'll be 4 soon, and uh, having a big career and being a mom, it's a lot. But I became a mom late in life, and so a lot of my career time was spent without a child, and to have a hard stop where I transition into mom mode and I'm just sort of in that bathed in that oxytocin love, I know that's important. I mean, I know from reading the science it is important, and I talk about it in the book, but I just feel it. So getting to having a hard stop in my work life now, and just getting to be a mom to this extraordinary little peanut, who's my daughter, is just a gift. It's like the deepest gift ever. So there's that. And she makes me want to live these principles because I want to be here with her for the long haul. I want to be here with her. I feel super passionate. I take this very seriously, very personally, because I started the parenthood journey later. So there's that. And I value meditation. So I'm type A, I'm driven, I'm doing a lot. Yes, I always have been. But again, the science is really strong that we need to shift out of this mind, even if it's just a moment at a time. And I take that seriously now. There's an app that I love. I'm fangirling on Healthy Minds. They're out of the University of Wisconsin, and they just put out this really cool app. It's free. You can have access to all of their teachings, all of their meditations. It's totally free. You don't have to unlock anything. You can give them a donation if you want to, which I have. And they research outcomes. They're researching the benefits of meditation. They have they have a meditation while driving. They have a meditation while exercising. They have a minute. They have a handful of these micro meditations. When I started my day today, I had there were a bunch of IT issues, and I had this podcast. It was kind of intense, and I just popped a micro meditation on, just remembering to breathe. Picking a word. Is it gratitude or is it calm or whatever? Just it's very helpful in moving out of sympathetic dominance. I eat, the principles of the younger you. I exercise. So, those are some of the things that are important to me.
How do mushrooms sort of fit into this picture, and what are your favorite, recipes or types of mushrooms that fit into this epigenetic kind of world?
I'm really bullish on mushrooms as very epigenetically important. And I think that their time has yet to come. Um, we're going to study beta glucans more carefully and the different types of beta glucans. And I think we're going to be able to see what kind of role, that they're playing. And I just have no doubt that they're going to be important. We know now that, I mean, mushrooms are almost like a corollary to liver in terms of some of them being just very micronutrient dense. And of course, you could throw them out in the sun and get some vitamin D, which I think is really cool. We actually have a nice vitamin D mushroom recipe that we popped in the book. Um, I like maitake, enoki. I like reishi. I like shiitake. Shiitake. We gave shiitake. We're considering one of our dynamic dozen, so we recommend it as a top um, nutrient in our program. But really pick any of those mushrooms, and I think um, they could also be in the dynamic dozen. We know that early studies show one of the ways that mushrooms exert their benefit, like in Alzheimer, for instance, is in changing DNA methylation patterns. Um, so they don't just have the methyl donor nutrients, which they do, and some of them have them in rich abundance, but I think that they have these methyl adaptogen properties as well in helping establish a favorable. So they're not just pushing methylation forward by throwing a bunch of methyl donors there, but they seem to be dictating a more favorable gene expression as well. Um, at least that's what the early research says. And as I mentioned, I think this is an evolving field, and we're going to have more exciting studies coming out, and probably we'll learn that some of the wonderment of mushrooms has to do with their ability to regulate gene expression. That's my belief.
Amazing. That's really cool to hear from your lens. So, where can people find out more about your work with your new book, with your research, and everything that's to come for you, and everything happening right now? Where can people find your stuff?
I have three things that I'll recommend. One is just younger.youprogram.com, younger.youprogram.com. The book is there. You can get a link to the app and learn about that. If you go to younger.you.com slash BASA, BASA, biological age subjective assessment, you can take our online biological age questionnaire. It's actually in the book, but you can also do it online. It's pretty cool. It's fun. It's helpful. It's not a validated questionnaire. We created it. Our team created it, and we worked with my biostatistician from our study to tighten it up. But I think it's useful. Maybe over time we'll be able to validate it because it is part of the study that we're going to continue to conduct. For my clinic and for sort of the broader Dr. Kara Fitzgerald world, you can just go to my website, which is just drkarafitzgerald.com, and you can get our blogs and you can see the clinic team, and there's just tons of content over there for folks and for professionals as well. So there's both regular layperson content as well as professional content.